Hello everyone and welcome back to my YouTube channel. Today in this lesson we will be understanding the protein folding and processing in the ER. As earlier described in my previous video, proteins are translocated across the ER and unfolded polypeptide chain while the translation is still in progress. So this video link given into the description box, please check and watch it. So here we gain the knowledge how protein fold it into the ER. So the ER is the major site of protein folding, glycosylation, addition of GPI anchor. So first is the ER is the site of protein folding. The first is protein folding, assembly of multisubunit proteins, disulfide bond formation, okay, which is only formed into the ER lumen, and linked glycosylation and the addition of glycolipid, okay, GPI anchor to some plasma membrane proteins. Now the polypeptide form into the three-dimensional conformation within the ER, assisted by molecular chaperone, okay, HSP70 that facilitate the folding of polypeptide chain. Now the polypeptides, okay, protein folding by chaperone. Okay, the protein folding in the ER by the chaperone. So in this figure, you can see the chaperone. Okay, this is the chaperone molecules. Okay, HSP17 BIP. Okay. So it thought to be bind with the unfolded polypeptide chain. Okay, so this is a polypeptide chain. Translation is still in progress. Okay, this is the polypeptide chain. This is unfolded polypeptide chain. This is it is crosses the plasma membrane and then mediate protein folding within the ER. Now this is chaperone helpful to the folding of polypeptide chain. Uh, Here also abnormally and improperly folded, okay, protein are targeted for degradation. So this is will be discussed in later. Okay. Now second one is the disulfide bond formation. Okay, so first, what is the importance of disulfide bond formation in the ER? Okay, so the formation of disulfide bond between the side chain of cysteine residues, okay, is an important aspect of protein folding and assembly within the ER. So this bond generally formed in the side chain of cysteine residues, okay, generally do not form in the cytosol because there is a reducing environment. In the ER, however, oxidizing environments that promotes the disulfide bond form. So now the how disulfide bond formed within the polypeptide chain. So disulfide bond formation is facilitated by the one of the enzyme, okay, that is a disulfide isomerase. Okay, you can see protein disulfide isomerase, this one, okay, protein disulfide isomerase, which is located in the ER lumen. Next is third is the protein glycosylation in the ER. So protein are also glycosylated on specific amino acid, that is the asparagine. Okay, so here it is the asparagine residue, and there is a glycosylation. As you know, the glycosylation, okay, just added oligosaccharide into the polypeptide chain. Okay, this process is glycosylation. There are two types of glycosylation, okay, N-linked and O-linked. Okay, so as earlier discussed in my previous video, glycosylation, types of glycosylation. So this video link given into the description box, so please watch it. Okay, so but here only ER lumen. Okay, this is the ER lumen. Only N-linked glycosylation occurs. Okay, so what happen in glycosylation? Okay, so oligosaccharide units, okay, there is a glycolipid, okay, that contain the oligosaccharide units, N is a glucosamine, mannose, glucose. Okay, so here is the anti-oligosaccharide is transferred from a dolichol carrier to polypeptide chain during the translation across in the ER membrane. Okay, translation is still in process, and the dolichol which is binds to the oligosaccharide with the N terminal of the aspargine. Okay. So now the next is the 3 glucose residues are removed from the oligosaccharide. Once the glycolipid carrier, okay, glycolipid carrier to polypeptide chain during their translocation across in the ER membrane. Okay, so glycol transfer the oligosaccharide. Okay, here N terminal of aspargine. Then after that, 3 glucose residues are removed. Okay, and then protein is modified. Then protein is modified for further process. Okay, and they transfer it to the Golgi apparatus. Okay, Golgi apparatus they transfer. So here, what is the importance of glycosylation? Okay, so glycosylation helps to prevent aggregation in the ER. Okay, and provide signals that promote protein folding for and subsequent sorting in the secretory pathways. Okay, secretory pathway, that means ER to Golgi apparatus, vesicles, and plasma membrane and cell exudates. Now fifth one is addition of GPI anchor. So some proteins are attached to the plasma membrane by glycolipids, okay, rather than the membrane spanning region of the polypeptide chain. So this is the glycolipids. Okay, this is the entire glycolipid structure. This is also glycolipid structure, and protein binds with the glycolipids rather than membrane spanning region. Okay, this is the membrane spanning region. Membrane anchoring glycolipids. Okay, this is the membrane anchoring glycolipids. Okay, contain phosphatidylinositol. Okay, this is a phosphatidylinositol, which also called the glycosyl phosphatidylinositol, which is called the GPI. Okay, so GPI anchor are assembled in the ER. Okay, this is the ER membrane, and added to polypeptide anchor in the membrane. Okay, by the carboxyl terminal membrane spanning region. Now the membrane spanning region is cleaved, okay, and the new carboxy terminus is joined to the NH2 group of ethanol amine. So this is the NH2, NH3 here is the NH2 group. Okay, this is the C terminal. The cleave is the C terminal of the polypeptide chain and binds with the NH2. Now which GPI anchor, okay, which contains the 2 fatty acid chain, fatty acid chain. Okay, linked to the inositol head group and oligosaccharide portion consisting of mannose and glucose amine residues. Okay, this is the inositol, and this is the mannose and glucose amine residues. Okay, glucose amine residues. So this is all about today's video, and you will get notes of today's video in the description box. And if you are new in this channel, please subscribe below and share this video. I hope this content will help you. Thank you for watching my video.